Hello, my dear students. Welcome to my today's online class on another topic in the phylum Protozoa as per your syllabus. After having learnt the various locomotor organelles encountered in the phylum Protozoa, you have to learn the mechanism or modes of locomotion as continuation of your previous chapter. Therefore, before going through this video lecture, better you watch once, recap the video lecture on locomotory organelle in the phylum Protozoa so that you will understand this chapter better. Okay? Now, let me introduce myself. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID on the slide. And after the class, for any queries, you can contact me. Now, I have divided this topic into two parts. Modes of locomotion part 1, which deals with pseudopodial locomotion or amoeboid locomotion, and second part where deals with the euglenoid locomotion and ciliary locomotion. So, to make you understand clearly and not to get into complications, I have divided this chapter into two parts. Today let us learn about amoeboid locomotion. Okay? Now, preliminarily you should know that basically there are four known methods by which protozoans move. They are, to list, amoeboid movement, which happens through the Pseudopodia, then swimming movement, which happens through either flagella, as you see in Chlamydomonas, Euglena, etc., or through cilia, as you see in Paramecium. Okay? Then, apart from these two major modes of locomotion, you do find gliding movement and metabolic movement. But as per your syllabus, you have to learn the first two types, amoeboid movement exemplified by Pseudopodia and swimming movement exemplified by flagella and cilia. The other two you are not learning as per your syllabus, but you should know that there are other modes of locomotion. In the study material, I have given some details about these two types also. Now, in this class, we shall try to understand the amoeboid movement or pseudopodial locomotion. Okay? This is the characteristic of the class Rhizopoda and exemplified by amoeba proteus and entamoeba histolytica. These exhibit amoeboid movement, and also such movements are seen in amoeboid cells. Okay? Amoeboid cells like macrophages and phagocytic leukocytes like monocytes and neutrophils of metazoans. So, apart from the protozoans, you do see this amoeboid movement in so many non-protozoan cells like macrophages, phagocytic leukocytes, etc. Okay? Now, various theories have been explained to date for giving a convincing explanation for amoeboid locomotion. Many have proposed, around 9 to 10 theories have been proposed. But of all these, the most convincing theory which explains the amoeboid locomotion happens to be sol gel theory proposed by Hyman. Students, you must have heard of Hyman series of zoology books. So, same person, lady, she is a lady, has proposed sol gel theory. I think now we have to study the sol gel theory in detail so that you can understand the mechanism of amoeboid locomotion. Okay. Sol gel theory convincingly explains the mechanism of locomotion involved in the formation of pseudopodia. Sol gel theory convincingly explains the mechanism involved in the formation of pseudopodia. Let us now study how exactly a pseudopodium is formed. Okay. This theory is also known as change in viscosity theory, which was advocated by Hyman. I have already told you. So, sol gel theory is also called change in viscosity theory. So, question may be asked by any of these two headings. Okay. And according to this theory, the cytoplasm of amoeba can be distinguished into two zones, outer ectoplasm and an inner endoplasm. I think this you already know, but outer ectoplasm is also called plasma gel and inner endoplasm is also called plasmosol. Don't interchange the terms. Ectoplasm is called plasma gel and endoplasm is called plasmosol. Okay. Remember this. Now, the plasma gel forms the outer layer of the cytoplasm. It is thick, more viscous, less in quantity, non-granular, transparent and contractile. You have to remember all these terms. Okay. It is thick, that means it is more viscous, that is viscosity is high, less in quantity, non-granular, transparent and contractile. Okay. On the other hand, the plasmosol, which forms the inner zone of the cytoplasm, central zone of the cytoplasm, is more in quantity, less viscous, fluid-like, more granular and opaque. Okay. Please note, let me come back to my previous slide. The plasma gel, which forms the outer layer of cytoplasm, is thick. Okay, that is more viscous, less in quantity, non-granular, transparent and contractile. Okay. Whereas the plasmosol is quite opposite to that. This forms the inner central zone of the cytoplasm. It is more in quantity, less viscous. Okay, less viscous, fluid-like, more granular and opaque. Please remember this. Then, due to change in viscosity, plasma gel and plasmosol interconvert. Okay, and consequently pseudopodia form and disappear, causing the movement of amoeba. Let me explain this in detail little later. You have to understand that there is plasma gel outside, plasmosol inside. I have already told you the nature of these two. But The change in viscosity, okay, may converts plasma gel into sol and plasmosol into gel, and this interconvert, okay, brings about the formation of pseudopodia and withdrawal of the pseudopodia, thereby causing the locomotion in amoeba. Okay, let's see that in detail. Now, this interconvertibility of plasma gel and plasmosol is basically a physico-chemical change. Gel becomes a sol by taking water, and sol becomes gel by losing water. How exactly it happens, I'll explain it to you. Then, to make you understand, this is a little complicated topic. To make you understand, the amoeboid locomotion has been explained in 10 steps, step by step. Let us understand what happens step by step, okay? Then, step 1. Initially, amoeba attaches itself to the solid substratum, okay? Initially, amoeba attaches itself to the solid substratum by plasma lemma at the temporary anterior end. Okay. Please note here the shape varies, right? So anterior end is also temporary, and at that region the amoeba attaches itself to the substratum. This is the first step, attachment of the amoeba to the solid substratum. Okay. Then step 2, the hyaline layer of the ectoplasm. I have told you there is ectoplasm and endoplasm, and in the ectoplasm there is a hyaline layer. What is this hyaline layer? It is a protein rich component. Okay, it is a protein rich component. This hyaline layer of the ectoplasm at the anterior end forms a thickened hyaline cap. I'll show you in the diagram. So the second step is, after the attachment, the hyaline layer of the ectoplasm. Note that hyaline is the protein rich component of the ectoplasm. At the anterior end forms a thickened hyaline cap. So this is step 2, first stage in the formation of pseudopodium. Then step 3, behind this hyaline cap, a point of weakness in the elasticity of plasma gel is formed. Please understand, attachment, first step, formation of thickened hyaline cap at the anterior end is the second step. Third step, behind this hyaline cap, a point of weakness in the elasticity of plasma gel is formed. Please note, plasma gel is highly elastic in nature, so a point of weakness is formed. That region, it becomes weak so that the sol can rush through, so that pseudopodium can be formed. That is the only concept here. Hence, the inner plasmosol flows forward, forming the pseudopodium. That is the reason why a weak point arises in the plasma gel. The moment it becomes weak, the less viscous plasmosol simply rushes in that direction and pseudopodium is formed. Okay. Then you can see here the diagram of amoeba explaining the sol gel theory. See, whatever you are seeing in the peripheral region, the dark blue color, that is ectoplasm or the plasma gel, and inside you see the light blue colored fluid, that is plasma sol. Okay. Then the advancing end is the place destined from place where the pseudopodium is destined to develop in this particular time, okay, in this particular picture. Then you can see in the ectoplasm, in the advancing end, okay, you can see a hyaline cap, that is the protein rich component, all right. And just behind this protein rich component, a weak point is created in the plasma gel so that plasmosol can rush in, right? So till now we have studied this much, right? Then let's see. Step 4. The plasmosol which flows outwards, okay, behind the hyaline cap, you can see the arrow marks there, changes its colloidal state from salt to gel and joins the ectoplasm. Alright. So it changes its nature from salt to gel and joins the ectoplasm. How? By losing water. Okay. It loses water. What is the reason for losing water? I'll explain it to you a little later, but remember. So step 4. Whatever plasmosol flows outward behind this hyaline cap at the weak point, okay, it changes its colloidal state. It transforms itself from salt to gel and joins the ectoplasm. How it transforms itself from salt to gel? By losing water, right? So, when water accumulates here, there is further growth of pseudopodium in that direction, okay? Then, step 5. The outer region of the plasmosol, which is flowing forward, undergoes gelation. It undergoes gelation and produces a rigid plasma gel tube, okay? The gelation of plasmosol, you can see here, zone of gelation, that dark blue regions, they are the zone of gelation. That is the region where, okay, the plasmosol loses water and becomes gel and joins the ectoplasm, and that zone is called zone of gelation. When this is happening, water is released, and because of the pressure, the pseudopodium further grows. Alright, the pseudopodium grows. Then, step 6. At this stage, when all this is happening, at this particular stage, 2 ends appear in the amoeba cell. The anterior end is smooth and round, that is the advancing end, where the pseudopodium is being formed, and the retractile end, also called wroid, which has a wrinkled surface. So at the step 6, you can make out clearly 2 ends. Anterior end where pseudopodium is being formed, and the posterior end, wrinkled end, that is the trailing end. Okay. This is step 6. Then again I have put the same diagram here to continue my explanation. Then step 7. Around the region of hyaline cap, here an annular region of salt to gel transformation is formed, and therefore this is known as zone of gelation. And please note at the opposite end, there is a region where the gel transforms into sol, opposite of what happens in the anterior end, and that is called zone of solation. Okay, zone of solation. This is the region where the whatever sol had become gel and joined the ectoplasm in this green colored areas. Here, again they absorb water, okay, and transform into sol. So you see the zone of solation, right, opposite to the zone of gelation. Okay. Then step 8. Plasma gel at the wroid end changes into sol. In the anterior end, sol transforms into gel by losing water, and that water, because of that water pressure, pseudopodium is formed. And at the opposite end, same gel transforms itself into sol by taking water. Okay. So plasma gel at the wroid end changes into sol and flows forward continuously. Students, you have to concentrate on the arrow marks through the gelatinized tube. As the plasmosol flows forward, pseudopodium elongates further and the body of amoeba moves in that direction. The ectoplasm does not move. Please note, ectoplasm doesn't move but grows at the leading tip and is broken down at the wroid end. This is what happens. Then, step 9. The gelation at the advancing end and the solation at the trailing end or wroid occur simultaneously and at the same rate, thus making the forward movement of the amoeba continuous. So this repeats several times and the amoeba moves, okay, in the direction where the pseudopodium is formed. Then, step 10. The contraction of the plasma gel at the trailing end causes hydraulic pressure, that is water pressure, on the sol and makes the plasmosol flow forward continuously in the plasma gel tube. Okay? Then, step 11. As the pseudopodium advances continuously in the direction of the movement, the body of the amoeba moves. So, this theory, which convincingly explains the formation of pseudopodium and the movement of amoeba, is known as sol gel theory proposed by Hyman. Then, all the time I was telling, sol transforms itself into gel by losing water and gel transforms into sol by gaining water. What is the basis? This may be a little complicated for you, but listen to the video lecture 2 or 3 times, you will become acquainted. Okay? Let's see now what is the basis. Why sol becomes gel, gel becomes sol, and how exactly it happens. Amoeboid locomotion is brought about by protein molecules. Okay? There are 2 protein molecules, actin and myosin. Same protein molecules are present in our muscle cells also. So, protein molecules present in the cytoplasm. Okay? They bring about amoeboid locomotion. Then, so the formation of interconversion of sol and gel, okay, phenomenon of gelation and solation was explained based on folding and unfolding of these molecules, based on folding and unfolding of these actin and myosin molecules. Okay, you can see this diagram where, according to these scientists who proposed this mechanism, okay, action of protein molecules, the cytoplasm gelates. The cytoplasm gelates means it transforms into gel when the protein molecules unfold by losing water. This happens in the anterior end below the hyaline cap. Okay. So the cytoplasm, that is endoplasm, that is plasma, the cytoplasm gelates, okay, sol, okay, gelates when the protein molecules unfold by losing water, and the cytoplasm solates, okay, in the opposite end when the protein molecules fold by absorbing water. So, in the zone of gelation, proteins unfold themselves and release water, and in the zone of solation, proteins fold themselves and absorb water. Okay. Hence, the proteins in the plasmosol, hence the proteins in the plasmosol are in folded state and the proteins in the plasma gel are unfolded state. Okay. Then same diagram I have put to explain. This folding and unfolding of protein molecules lead to the formation of the pseudopodia and thus the amoeboid movement. And the energy required for this process comes from the ATP, that is adenosine triphosphate. Then there is one more theory, okay, foundation zone theory put forward by Allen. Plasmosol flows forward due to the pulling force caused by sliding action of actin molecules over the myosin molecules at the advancing end. Okay, but it is enough if you understand that interconversion of sol and gel. But as an additional information, you can know this. Okay. So the interconvertibility of sol and gel is mainly due to the assembly and disassembly of actin filaments. Assembly results in gel formation and the disassembly results in sol formation. Okay. So it is the basis for the interconvertibility of sol and gel finally goes to the credit of proteins, okay, actin and myosin. Dear students, so in these slides I have tried to explain the locomotion in amoeba by the formation of pseudopodia, how exactly the pseudopodium is formed and what is the basis for the solation and gelation. Study the video lecture 2 to 3 times so that you will understand little complicated concepts well. Thank you for your patient listening. Any queries you can contact me. Till then, stay blessed.